Hi everyone. So just a first introduction to the Intraco group. So Intraco was funded through a joint call from SDIV, the Synthesis Center of IDIV and CESAB. And this synergy joint call funded a smaller group than usual. And we had workshop both in Germany and in Montpellier, France. And the group gathered people from France, Germany, and the US mainly, and Australia, sorry. So at the center of our project was the observation that communities in nature can shelter an amazing species diversity. So for example, this is illustrated in this image from a tropical forest canopy, which can actually shelter more than 100 different tree species within a single hectare. However, despite decades of research to understand this amazing diversity, how those different species can stably coexist remain quite unclear. And the fact that this is both widespread and both difficult to understand is called in the literature the coexistence paradox. And currently, global change actually emphasize the consequences of this coexistence paradox is that models remain unable to simulate diversity without an unrealistic assumption. So by unrealistic assumption, I mean, for example, assuming an external seed rain or assuming perfect trade-off among species that are actually not observed in data. However, being able to simulate diversity realistically is critical to predict global change effect on ecosystem functions and diversity. So if we have a closer look about how the coexistence paradox has been tackled in the literature, most hypotheses and tests have been so far developed under the assumption that different individuals within a species are fully identical. So this was typically made, for example, by using species mean attributes such as performance or traits. However, data showed a potentially important variability among individuals within a single species. So this is, for example, results from a meta-analysis published by Seifert and collaborators in 2015. And what you can see is on the y-axis, many different plant traits, and on the x-axis, and on the y-axis, the proportion of intraspecific variability in the total trait variation within plant communities. And as you can see, such intraspecific variability can represent more than half of the total plant trait variation within communities. So our goal was, so the question now is, what is the role of this intraspecific variation in species coexistence? And over the past 2 decades, a range of studies have attempted to answer that question. But however, they reach contrasting conclusions. So some of them concluded that it slows down competitive exclusion and thus foster coexistence. But others concluded that it prevents coexistence, and some found contrasting results. So in this context, our goal was to revisit the role of intraspecific variability in species coexistence. So in the first part of the project, we tried to look at the sources of intraspecific variability and how these potentially different sources of variation can have different consequences on conspecific similarity. So the question is thus, where does observed intraspecific variability come from? So let's take 2 species, species 1 and species 2. And on this plot, you can see on the y-axis the performance of those 2 species in an environment E0 characterized by a unique environmental variable X. And on this plot, conspecifics, so individuals within each species, are identical. And what you can see is that species 1 outcompetes species 2 in this environment E0. So the observation, what I just showed you from the meta-analysis from Seifert and collaborators, is that actually data shows a range of value for each species in E0. And there can be 2 interpretations of this observed variation in performance or any proxy of performance in a given environment. So the first one is that conspecifics are intrinsically different, and with the immediate consequences that some individual from different species, for example, this one and this one, can be more similar in performance than some individuals of the same species, for example, this one and this one. And potentially, this variation can be due to genetic variation. A second interpretation of this observed variation is a result from the fact that potentially E0 was mischaracterized and there is some unobserved environmental dimensions that can actually influence individual performance in E0, but that we actually miss when measuring the data. So if we now pushing forward this second interpretation, if we plot performance in a two-dimensional environmental space, so you have the X environmental variable on the X-axis and the Y environmental variable on the Y-axis. What we can see is that contrasting performances among conspecific individuals in E0 actually do not result from intrinsic differences among them, but from differences in the local environment they actually experience, and that was previously poorly characterized. So if we then go back to the one-dimensional perspective we had at the beginning, we can see that the apparent niche overlap in one dimension come from actually an absence of niche overlap in two dimensions and exclusively result from the projection of the two-dimensional space in one dimension. These two interpretations are, of course, not mutually exclusive, but what is important to have in mind is that they can have different signature in space and time. One can be unstructured, but especially if we remove potentially dispersal limitation in space. And one can be highly structured in space and time because environmental variation is typically strongly spatially and temporally autocorrelated. And both have very different consequences for conspecific similarity and thus for coexistence. So to try to tease apart among these two interpretations, we search for evidence in different types of data. So first, we just have a look at how environmental variation is documented in the literature. And actually, there is a growing evidence that environmental vary in many dimensions that are potentially independent from each other. And those environmental dimensions can vary at very different scales. So here you have just a few examples, such as soil nitrogen content, soil water content, different soil elements, or topography. So the conclusion of this first observation is that environmental variation is high dimensional and multi-scale. Then we looked at data from forest inventory across 3 natural tropical forests. And we look at the proportion of individuals within those communities which belong to species with significant spatial autocorrelation in growth. And actually what you can see is that across these 3 sites, those individuals represent a substantial proportion of the total individuals within communities. And similarly, the proportion of individuals belonging to species with significantly smaller intra versus than specific semivariance was high. So to conclude on this point, individual growth is largely spatially autocorrelated across 3 tropical forest sites. So here growth was taken as a part of what builds individual performance. Finally, we also look at data from an experimental clonal eucalyptus plantations. And this data was used because we believe that it represents an extreme case of controlled genetic variation. So different eucalyptus clones are planted within the site. So genetic variation is fully controlled. And the setup was so that the environmental variation was minimized. And what we found is that the variability between individuals within clones, so between individuals sharing the same genetic, that have the same genotype, was almost twice as high as the variability between genotypes. And together, these different evidence show that Large intraspecific variability can emerge from high-dimensional environmental heterogeneity. Intraspecific variability actually does not necessarily imply that individuals are different, nor that species niches overlap. And intraspecific variability is likely structured, is for a great part structured in space and time. And despite intraspecific variability, conspecific answer more similarly than heterospecifics. And together, this has important consequences for species coexistence. Yeah, sorry. And in the second part, we go back to the modeling studies I showed you before to have a look at how IV was actually represented in those models of community dynamics. So basically, intraspecific variability in those models are including using a random noise around species means. So any individual within species is given attributes that is drawn randomly around the species mean. And using a virtual experiment, we try to explore the effect of using this random unstructured intraspecific variability as a substitute for environmentally driven structured intraspecific variability in a coexistence model. So let's imagine you are in a virtual world with n independent spatially autocorrelated variables, so E1 from E1 to En. Those n environmental variable describes the environment in a d time d cell grids. Then let's imagine we have S species whose performance is fully described by n independent optima. Each optima corresponds to the optimum along one of the n environmental variables. So this describes the true performance as a function of those n environmental variables. So this leads to, in the end, a matrix of d time s dimensions, where you have across all the sites within the grid the performance of individual of each species. Now let's imagine that actually among those n environmental variables, we correctly characterize, quantified, and observe only one of these variables, E1, while the rest of the environmental variables are actually unobserved. Then what is typically done, so those data on the right are typically the type of data you can derive from forest inventories. And these are typically the data availability we have on the environment in this type of settings, in this type of empirical settings. And typically what we then do is a statistical inference where we do have the true performance from the forest inventory, and we try to understand how the environment characterized only along one variables drive this true performance. So beta 0, beta 1, and beta 2 corresponds to the effect of the first environmental variables. But then the residual of such a model corresponds both to potentially measurement errors, but also to the effects of the other environmental variables that are actually unobserved. And from this residual, we can derive some variance that we can estimate. We can estimate it. Then there are two ways to then use this model to predict performance. The first one is just to remove the residual variance that was unexplained. And this is what I will call in the next few slides the partial knowledge model without intraspecific variability. Then another approach would be to actually use the variance you have derived from your statistical analysis and add a random noise to the performance estimated using the beta parameters. And this is what I will call the partial knowledge with unstructured IV. Then we compared the communities simulated with the same simulator of community dynamics, but using the three different performances. So the one that is actually the true one, the one that we assume can be measured in the field, or the one predicted using the partial knowledge with intraspecific variability, or the one predicted with the partial knowledge model with unstructured intraspecific variability. And so what you can see here are results from a simulation with these three performance model. So on the x-axis, you have the number of observed environmental variable. So in the previous slide, I only provide the example where only one variable is observed, but we replicate the experiment with until 9 observed variables among a total of n environmental variable of n equals 10, environmental variables. And on the y-axis, you have the species richness of the final community simulated using these different predicted performance. So for each number of observed environmental variable, you have on the left output from the species richness drawn from the simulation using the partial knowledge without IV. And on the right, you have output from the simulation using the partial knowledge with unstructured IV. And what you can see is that partial knowledge models actually never reach the level of the true performance model, which you can see on the far right. But what we can also observe is that starting from two observed environmental variable, adding unstructured variability actually lowers species richness each time. So the take-home message of this small virtual experiment is that adding randomness, as it's majoritarily made in a modeling study that attempted to explore the role of intraspecific variability in species coexistence. So adding randomness does not compensate for the lack of knowledge of the environment. And unstructured IV is actually a bad surrogate for unobserved environmental variation.
Okay, so these main results have been published in two papers from the PhD thesis of Camille Gérard-Tercieux, so in the framework of the Intraco model. And now I will try to discuss about a simple mechanism to explain the stable coexistence of a high number of species. So what Isabelle has shown us is that you can have a high intraspecific variability, but it does not mean that individuals from the same species are intrinsically different. And so we have shown that intraspecific variability can emerge from the high dimensionality of the environment. And that finally, IV is not a direct cause of species coexistence. So in fact, the question of the stable coexistence of a high number of species is still open. And so we are wondering if we can provide a simple theoretical mechanism explaining this coexistence of a high level of species, of a high number of species. And we wonder also how intraspecific variability is related to this mechanism and how to integrate this potential mechanism in empirical community models so that we are able to simulate community with high diversity. So in fact, we try to show using a theoretical model that coexistence emerges from the high-dimensional interaction between species and their environment. And it also emerges from the fact that species are different in a high number of dimensions and that this difference act on their answer to the environment, on their response to the environment. And so this is not really new, in fact, and That's only the niche theory which was proposed by Hutchinson more than half a century ago. But what will be new in our study is that we will show that we can model this high dimensionality in community models using a species covariance matrix when all the dimensions of the environment are not available. So to show that, we used a theoretical model where we consider a landscape with K sites and S species. And so on the different sites, you can have several individuals from different species which are competing for resources. And on each site, you have a particular environment defined by a given number of dimensions, so E. And for the demography of the individuals, we considered a combination of dispersal, performance, so performance, for example, would be growth or fecundity, and survival. What is important is that dispersal is the same for all species, and survival also. So dispersal is a fixed fraction of performance for the individuals and is uniform on all sites. And for survival, it is density dependent. So because species are competing and individuals are competing for resources on each site, you have a survival probability which depends on the density of the individual on each site, and this survival probability is the same for all species. And regarding the performance, so the performance will differ between individuals and will be modeled using 3 different approaches that I will detail in the next slide. So we consider the performance of each individual. So it is the performance J. So for individual I of species S on site K I time T. So it's really an individual performance. For the first model, that is denoted SK for structured and known. In fact, the performance is derived from the species parameters and the environment in a high number, supposedly, of dimensions. So we will make the number of dimensions vary to see what will be the impact on the community. And in the second model, so denoted SU for structured unknown, the performance of each individual depends on a species covariance matrix, sigma, which is directly derived from E and S. And then for the third model, so this second model is in fact structured by the species covariance matrix, even though we don't know the source of variability, because we don't know, in fact, precisely the dimension of the environment for the simulation. In the third model, so we considered an unstructured and unknown model where performance of each individual is random in this case. And these are the results we obtain using these models. So in our simulation, we observed that if E is large, so let's say much greater than 1, only the SK model where you have the interaction between the species parameter and the environment in a high number of dimensions, and the SU model where the individual performance is defined by a species covariance matrix. Only in these 2 cases, you reach a stable coexistence. And in the case of the UU model, where individual performance is completely random, you don't have a stable coexistence. So we can see that on the figure on the right, where you have 2 cases. At the bottom, you have the case where the environment is defined by 2 dimensions, and the case at the top where you have the environment defined by a high number of dimensions equal to 100. So in the case of the SU and SK model, you can look at the abundances of the species with time, and you can see that the abundances are stable and that several species are coexisting. While for the UU model, where performance are random, in fact, a limited number of species can coexist, and you have a random drift, so some species are excluded quite rapidly from the community, and you end up with a low number of species coexisting. What we show also is that as soon as the number of environmental dimension is high, what happened in the case of the SK and SU model is that you have a strong diagonal dominance in the species covariance matrix. So, for example, on the figure on the right, you can see that for a number of dimension of 20 on the right, you have a strong diagonal dominance for species covariances, while it is not the case when the number of dimension is low. What does it mean when you have a strong diagonal dominance? It means that, in fact, intraspecific correlation are greater than interspecific correlation, which means that individuals from the same species, what we call conspecific individuals, they respond more similarly to change in the environment than individuals from different species. And so what happened in this case is that competition concentrates within the species, which is a necessary condition for species coexistence, as shown, for example, by the work of Ghislain in the years 2000. And of course, the SU model with the species covariance matrix is not perfect. And only in the case of the SK model, where all the environment is known in a multiple number of dimensions, shows a species sorting according to where they can dominate. And so we can observe a species sorting on the different sites as a function of the environment. So the SU model is not able to explain all the outcome of the community dynamic, but at least it is able to make species coexist and to maintain a stable coexistence of the species. Here we can have a look at the impact of the dimensionality of the environment on the number of species coexisting. So we can see that we reach a high number of coexisting species. So on the figure on the right, where you have the species richness as a function of the environmental dimension E, so from value, for example, 5 to 500. So you can see that the number of species coexisting is much higher than the number of environmental dimension. So for example, you reach about 50 species coexisting as soon as the environmental dimension is greater than 5. So the number of coexisting species quickly increase with E. So to conclude, we can see with that model that coexistence can emerge from high-dimensional E cross S interaction. And in fact, the coexistence is obtained without considering tight trade-offs in the model. So we are not saying that there are no trade-offs in nature. Of course, there are trade-offs because species are, and in trees, for example, are living organisms that need to follow the physical and chemical rules. So there are trade-offs. A species cannot be performant in every environment. But here in our model, we do not constrain the species to follow specific trade-offs. So species parameters are drawn at random, in fact. And so this is a real important output of our work. Also, we show that high dimensionality in the environment is important. And in fact, this does not mean that you have a high number of resources, for example. So you can have a low number of resources, such as light, water, nutrients, but you have also either environmental factors acting on individual performance, which are topography, soil, climate. So in fact, the dimensionality of the environment is high by nature. And also, resources can be divided in bins. So for example, you can consider an individual performance at high or low level of lights. And so this increases naturally the number of dimensions of the environment. And so in fact, we argue saying that stable coexistence can be easily achieved when you consider the high dimensionality of the environment and the species differences in a high number of dimensions. So what we want to underline is that to understand coexistence, in fact, we should not think in terms of intraspecific variability as intraspecific variance, for example, but we should think in terms of covariance between individuals locally, and to make sure that individuals from the same species respond more similarly to the environment than individuals from different species. So this is the first conclusion of this theoretical work. So I think we can have more time to present more recent result about our Intraco group. So first, these results have been presented in a preprint available online. So which are the result of this theoretical work. And we are trying to test, in fact, if we can observe this diagonal dominance in forest at the global scale. So what we have done is that we have compiled a global database using forest inventories with at least 4 consecutive censuses in different forest biomes, so including tropical forest, but you have also temperate forest, subtropical forest, and boreal forest. And we have compared intra vs interspecific correlation for tree growth and fecundity at the local scale. So within the inventories, considering subplot of 20 by 20 meter, for example, where competition occurs. And what we have found is that either for fecundity or growth of individual, we have observed that all correlations were positive. So this means that, for example, when you have a change in the environment, all species tend to respond to this change in the environment in the same direction, which makes sense because, for example, if you have higher temperatures or if you have a higher level of light following logging, for example, all species in general will have a higher growth. But what we are observing is that in most of the cases, so in a high proportion of comparisons, intraspecific correlations is greater than interspecific correlation. So again, intraspecific individuals respond more similarly than individual from different species. And we have shown that this was the consequence of having a stable coexistence of the species. And we observed that in the data. So in fact, we observed the diagonal dominance necessary for species coexistence in all forest biomes. Then to conclude, and it's a work in progress, we are trying to develop approach to include the species covariance matrix in empirical community models so that they can generate diversity without using artifacts such as a seed rain, for example, in forest community dynamics model, in forest dynamics model, sorry. And so how to do that? So I'm sorry, here I just have some text, but in fact, what we want to do is to try to infer demographic mechanism, demographic processes using some explanatory variables and try to estimate residual species covariance matrix from the residual. And so combining, in fact, the fixed demographic processes with the residual species covariance matrix, we can obtain, in fact, models of forest dynamics or community dynamics, which could be used to answer some specific question, for example, on the effect of climate change on species diversity and species response to drought, for example, but also or while being able to generate diversity and being realistic. Okay, thank you for listening. So I will stop the screen, and we are open to question if you have some.
Yes, thank you very much. That was really interesting. So yeah, we have time for questions. So if anybody has a question, please raise your hand and I will distribute the floor. So we have Melanie. Hello.
Hello, sorry, I'm from the lab, so happy to hear the results, and I was wondering at what scale does this competition occur? Maybe I missed the information. Is it really like a 1 hectare scale, or at which scale? Yeah.
Okay. Should I answer? Yes. Yes. So in fact, we tested different scale. But so at first we consider a small scale competition, so about using subplot of 20 by 20 meter, and so we consider the correlation at this scale, but using also different size, so using data on a 1 hectare plot, so 100 by 100 meter, we also observe that intraspecific correlation are greater than interspecific correlation.
Okay, well, thanks. It was raising question about networks analysis that also often erase the intraspecific variation that do detect a lot of exclusion between species. But I think I will speak with colleagues working on networks because or share perhaps your contact, because I think they erase this variation while they could detect and have also often data to detect this exclusion.
Okay.
Well, thanks.
Thanks, Melanie. Henrik?
Hello, can you hear me?
Yep.
Yeah, thank you very much for your presentation. I'm not an expert on the topic, so maybe it's a very naive question. But if you think about the modern coexistence theory, where we have that coexistence is explained by differences in niche differences and differences in competitive interactions, antagonistic interactions among the species, the fitness differences among the species. Do you think that your framework could somehow include this kind of differences if you have, for example, different access to light of different species, or some species that have a higher fitness, they reproduce more than others, or something like that? Maybe it's totally nonsense, the question, but
well, I think you can include any kind of mechanism and processes in the model, as soon as you make sure that you include also a structure in the residual covariance between species and individual. Otherwise, usually you won't work in a sufficiently high number of dimensions, and you won't reach species coexistence. But you can include, yes, many different processes showing species differences or including species differences in a high number of dimensions, yes.
yeah.
I don't know if it answers to your question.
I guess you won't have the data, right, to compare these intraspecific fitness differences, right, for most of the plants that you have in your data set, for example.
You mean trying to reproduce the analysis, comparing intraspecific and interspecific correlation, but using other processes?
Yeah.
Yes, for the moment, it was easy to access and to have data on tree growth and tree fecundity using a forest inventory plot. And it's already good because having data, for example, on fecundity is not always available. And so it was good to be able to show that on these two, I would say, main processes which explain species performance in different environments. But for other processes, we could think about other specific or precise experimental setup to see if it works or not.
Thank you.
I think Melanie has another question. Oh, you're still muted, Melanie.
No, sorry, I had another question about how far were the differences, or did you include it, for example, some species that were usually monodominant, perhaps from Europe or North America, or some hyperdominant from South America? Is this trait also impacting the results you had, or?
Mm-hmm. Okay, so Isabelle, maybe you can answer now if you
No,
have.
you can.
No?
Okay.
So that's a really interesting question, because in fact, what we want to show with our work is that the question should not be how to explain coexistence, but the question which should be in fact easy, because the environment and the species difference is in a high number of dimensions, and we can see coexistence, species coexisting everywhere. But in fact, the difficult question should be how to explain monodominance of some species in some places. And so we are trying to see how our framework can explain monodominance of species in some places. And so, for example, it could be due to the fact that the environment is strongly constrained, and that in fact in this and so the number of environmental dimension is low in fact in this case, and that only a few number of species are performant in this typical environment. So we need to find data, in fact, to show that this framework is also able to simulate monodominance in some particular case and some particular environments.
Okay. If I may, because I work more on fungi that often are assumed to be a factor favoring the monodominance in the way they reduce somehow the competition because they favor intraspecific interactions. So I would be really curious whether either fungal interaction and the way they favor monodominance do interact with perhaps the reduction of intraspecific competition.
But I think that in our framework, what you just say is that the fungi interaction actually impact the environmental dimensions and the number of them and their variation.
Yeah, they may increase the number of variation, that's for sure. So that's a bit counterintuitive if you think about monodominance restricting the dimension to favor intraspecific, reduce intraspecific competition. That might be the contrary.
Okay, I see your point. Yeah.
Well, to be discussed, but
yeah, to be discussed and to be tested. Yeah.
Right. But there, I think that if you know your species list, it's easy to know which kind of interaction they do have as a micro-result traits. So, and sometimes it doesn't support the monodominance. I think with at INFIOL, we were contrasting the distribution patterns with the ectomicro-result interaction, and did not work in Africa, for example. So, but as you have a lot of data, that might be a trait to be considered.
Okay, thank you.
Well, thanks.
Are there any other questions? I have questions, but I will sacrifice myself if anybody else has them in priority. But if not, then I might just go. For me, I'm also going to ask quite naive questions, I think, so excuse me in advance for this. But so if I understood correctly, I took notes and everything, but the diagonal dominance, it means that the species that are from, so that there is less intraspecific variation than interspecific, right? So it means that species that are closer genetically, they tend to respond in the same way, right? Correct? Which is what we would expect. And so at some point in those last results that you were showing, you said that this diagonal dominance is a consequence of species coexistence in your model. But wouldn't it be the other way around? Or is it just in the way that you're thinking about things and, like you were saying before, things that you're trying to show, and it's not necessarily the reality, but just like with how you modeled things? Because that kind of stirred me when I heard it. Am I being clear?
So in the last part Ghislain showed, I think our view of this is that the diagonal dominance is a signature of mechanisms supporting coexistence widely.
Okay, so that you can't have species coexistence without having this diagonal dominance, right? But this is within your model. Okay.
And in the model, we use this as a sort of trick to structure the absence of knowledge on underlying mechanism in ways that they still support coexistence.
Okay, I see. But it's like, okay, so it's like a trick
right that we
to like
use
parameterize. Okay.
in the two ways.
Yeah.
Like in the last part, it was more consequences of mechanisms supporting coexistence, but then in the model, we assume that potentially using this could support coexistence in case we do not know many things. So this is in both ways.
I see.
In fact, it's following all the work by from the Lotka-Volterra model or from Chesson, which is showing that to reach stable coexistence of species, you need a stronger intraspecific competition than interspecific competition. And in fact, the diagonal dominance is just that.
Yeah, okay, I see.
Means that conspecific individual respond more similarly to a change in the environment than individual from different species.
Which is kind of reassuring, right? Like this is what you would want.
Yes, this is in fact something which is quite obvious.
But that you want to show and what you want to have this mathematically in order to go beyond, right?
Yes, and even though we know that, we all know that, that models are still not able to maintain diversity and simulate coexistence easily without using some artifacts. So the question was, what mechanism should we include in empirical model to maintain diversity when we don't have access to the environment in all its dimensions, in fact. And so in the first part, we are showing that using only intraspecific variability with a random term leads to wrong outputs regarding the coexistence of the species. But it leads to, if we use not the variance, but the covariance between individuals, and with a strong diagonal dominance, in this case, we reach a stable coexistence of the species.
I see. I think. but no, but I think I do. It's good. It's been a long time since I haven't studied these things, but I always really enjoyed it. Melanie, is that another question, or is that you just forgot to unraise your hand and I didn't follow?
No, well, sorry, I'm in the lab in the meantime. No, there wasn't any more question. I forgot to lower the hand.
Okay. Sorry. So if there is no last question among the audience, I guess we can call this a session.
Okay.
And
but
thank you very much.
Thank you all for attending.
Yep.
And if you have questions, don't hesitate to reach us and to discuss about these findings. Yeah. Thank you very much to people at CESAB also for offering us to present this work.
Yeah, and thanks for accepting to play along. It was lovely.
Okay.
Bye.
See you.